This video is on how to do the histology for the respiratory system lab. We are going to do the nasal epithelial, we are going to do the trachea, and we are going to do the alveoli. The nasal epithelial is the simplest, and I like this just because it shows the layering that you get. Now in the nasal epithelial, and in actually all the tubes we'll see here on out, you have at least two layers, usually a third layer. The first layer is called mucosa. The mucosal layer is lined up against wherever the lumen is. So the lumen would be here. The submucosa is underneath the mucosa, hence submucosa. And this tends to have blood vessels, so there's a nice vein. There's an artery, so see the artery is thicker. There's nerves, there are glands. That's might be a gland right there. And then for the intestines and the urinary system, we'll have a muscularis. Then you have an outer layer of connective tissue. Some drawings I saw online actually put the bone and the cartilage in the trachea as part of the submucosa. I don't have an opinion on that one way or another. I was taught the submucosa is just the connective tissue underneath. So let's draw this. So we're going to start with the ethmoid bone. And I want the ethmoid bone on the top. So here's my ethmoid bone. And then down here, I'm going to draw my mucosal layer. So that is pseudostratified. So let's draw some pseudostratified cells here. Now it's ciliated pseudostratified epithelial, so I need to put cilia on. So I'm going to put a little cilia on all these cells. The mucosal layer also has these cells called goblet cells that are supposed to look like a wine goblet. And let's see if I could draw goblet cells. I cannot draw goblet cells today. So we have these goblet cells. So let's label all that. So we have our mucosal layer, we have cilia, these are little cilia, we have our goblet cell, and we have our pseudostratified epithelial. Then we have the layer of bipolar cells, and that's fun. It's just doing a bunch of black dots. Now, I like this histology slide because it shows the vessels as well. So we are going to have an artery with its thick muscularis, and we're going to have a vein. Remember, veins are larger, but they tend to be amorphic, with its thinner muscularis. Now, there are these glands in here called Bowman's glands, and they actually add to the secretions of these goblet cells. So this is the submucosal. I could put a nerve in there. Let's put a green nerve in. And that's basically what the epithelial of the nasal cavity looks like. So let's move on to the trachea. For the trachea, if I was really smart, I would just take my pen tool, circle this all, and make it go away. But I'll do it from scratch. So the trachea slide is going to have this C-ring cartilage that sort of looks like the letter C. So there's the C-ring cartilage. And then I'll just have connective tissue. It's sharing with the esophagus here. So the esophagus would actually be over here. Now, since the trachea is cartilage in its outer layer, we have to put in our lacuna. And we're not going to worry about the layers of the esophagus for this drawing. That would be the muscularis, by the way. Now, we have a layer called the submucosa underneath that is mostly areolar connective tissue. So I'm going to just draw in the submucosal layer. This is going to have arteries and veins. So let's draw some nice round arteries with a thick muscularis. And then some amorphic veins with a thin muscularis. So, artery, vein. Now there's some submucosal glands, so let's just draw in a couple submucosal glands. Yes, I like the glitter pen, I don't know why. And let's do a nerve. There's a nerve. So this is the submucosal layer. Oops. Then our final layer, we have the mucosa, which is going to be these pseudostratified epithelial cells. And again, it has goblet cells and cilia, so let's put in a goblet cell. And I'll draw them how they actually look on the slides, usually not nice goblets. There's room for a goblet cell. And that's our cartilage side. So let's see how I did. So here's our goblet cells. See, they do look like blobs a lot of times. They really do, if you get a nice one, look like goblets, though. We have the cilia in our pseudostratified epithelial. We have our submucosal layer. There's a vein. Remember, veins are amorphic things, so that would be the vein right there. I don't see an artery. This is a duct for one of the submucosal glands. Then we have our hyaline cartilage. And again, I'm throwing this in as a part of the submucosa. I've heard it's part of the submucosa. I've heard it's not. But since your textbook had it as the submucosa, I'm putting it in. Then the outer layer is called the adventita. I'll do serosa when we do digestive system. They are different. There's basically no fluid-filled cavity on the outside. That's the difference. But this is what the trachea looks like. Our next slide is the lungs. And in the lungs, we are going to have lots of tubes. And we're going to have bubbles. This would be an alveoli. Alveoli are circular sacs at the end. This would be an alveolar duct. The alveolar duct is a tube, but here's another one. It doesn't have a muscular layer. This is a bronchiole. It has a thicker mucosal layer, and then it has a muscle layer in there. And then the bronchi, which I'll draw on my slide just for the sake of drawing it, will actually have a ring of cartilage. So, I'm going to assume you're looking at this on a power that you can't really see all the cells. And we're going to have these closed end sacs. These closed end sacs are called alveoli. So let's draw some alveoli down here. And they tend to all be connected to these alveolar ducts. And if I want this, so here are the alveoli, here are the alveolar ducts. If I want this to be real moralistic, I just draw some random alveoli and some random alveolar ducts, but I want room for other structures. So the alveolar ducts are going to increase in size, and they're going to merge into these structures called bronchioles. Now the difference between the alveolar duct, which is this, and the bronchiole, which is this, is the bronchiole is going to have some muscle tissue with it. So I'm going to put on some muscle there. So see, this has muscle tissue with it. And these guys I'm going to attach. And there'd probably be more bronchioles, but again, I need room for other structures. Now the bronchioles, let's draw another one coming in here, just from outer space, I guess. So the bronchiole, and let's do another one coming in there. There's my other drawing. So the bronchioles have a muscular layer. These are going to merge and form something called bronchi that are going to have a layer of cartilage. So, and this is going to have pseudostratified epithelial, and there's a layer of cartilage. Oops, too big. So there's a layer, instead of the muscle layer, there's a cartilage. So bronchi are bigger than bronchioles. They have cartilage. The bronchioles have muscle. Very important difference. And, of course, they will have blood vessels. So we've learned the artery is going to be this bigger muscularis, and it's going to hold its shape, while the vein is going to have a smaller muscularis and be this amorphic blob. And if I actually drew this correctly, the artery and the vein would actually be over by the bronchi. So my artery and my vein are actually over here by my bronchi. And that's pretty much what it looks like. Here's our bronchiole with this muscular layer. There's our alveolar duct. The vein's going to be more amorphic. The artery is going to have a thicker muscularis and hold its form. So those are the histology slides. Now, as part of your follow-up, you're going to label all of this. It's a lot, I am sorry. So, that's how you do the histology. Have a good day.